Hi, this is a recording about gene expression. It's an overview of the process. The process is taking DNA, the genes in DNA, and ending up with a product that's useful. Frequently, a protein is what we talk about, but it could also be regulatory RNAs. When we talk about gene expression, there's a process called the central dogma, which has actually changed over the years. And so far, or at this point, here's the kind of idea we have. So we start with DNA molecules, which remember are made up of DNA nucleotides. And then those are read, and they allow the construction of RNA molecules, which are made of RNA nucleotides. And those nucleotides, remember, the RNA and DNA are nucleic acids, that type of large molecule group. Then the RNA is read, specifically the mRNA, and that codes for certain polypeptide combinations. So the polypeptide is made of amino acids. And then the polypeptide ends up folding and getting modified, maybe coming together with other polypeptides, and it becomes a protein. And the protein has some sort of function in the cell, whether it's an enzyme, catalyzing reactions, or a structural protein, storage protein, a receptor in the membrane, many different functions there. To get between these different molecules, there are different types of processes that occur. So the DNA to RNA, that process is called transcription. I abbreviate that TXN. The process between RNA and the polypeptide is called translation. Sometimes you'll see that TRSL. The process between polypeptides and the proteins, that's folding, the polypeptide folding and processing. And basically, we're constructing the tertiary structure of the protein between the polypeptide and the protein, possibly also quaternary if there are multiple polypeptides coming together. The way we think about that would be if there are multiple subunits. Quaternary structure. You should recall those things from the lesson on macromolecules. So looking at that as our overall process. There are different molecules involved in each of the transitions. I'm just going to lay those out now. We'll talk about them in more detail later. The transcription process, the DNA to RNA, the molecule that allows that to occur is called RNA polymerase. When we talked about DNA polymerase, when we discussed DNA replication, RNA polymerase is going to build the RNA pretty much in the same way that DNA is built, using DNA as the template to match up complementary base pairs and build the RNA strand. Let me just mention that the RNA polymerase makes all kinds of RNA. It can make the RNA that's taken and read to make polypeptides. That specific type of RNA is called messenger RNA or mRNA. But there are other types. There's also tRNA. That stands for transfer, and that's going to be important in translation. There's rRNA, which is ribosomal RNA, also important in translation. It is a part of the ribosome complex. There's siRNA, small interfering. This is used for gene expression regulation. In certain organisms, the siRNA, which are these very small RNAs, are going to be able to turn off gene expression by regulating other RNAs. Similarly to siRNA is miRNA or microRNA, also gene expression regulation, turning genes off by interacting with the mRNA. And then finally, the one we're going to discuss, piRNA, which actually stands for Peewee RNA, and this one is important in stem cells. It's also a regulatory, very small pieces of RNA that are transcribed by RNA polymerase as well, but don't get translated to make polypeptides. So RNA is a very versatile molecule, as you can see, does a lot of different things. Many of these, the small non-coding RNAs, meaning that they don't code for polypeptide, are relatively new as of 2014. I mean, we've known about them maybe for, 10 or 15 years, but relative to knowing about DNA and mRNA, it's been a recent development. And so we're still working on understanding how those help with gene regulation. And we think that a lot of our genome is coding for them, and we kind of had ignored it up to now. And so that's a big area of research that's pretty exciting. In this process for eukaryotes, there's an additional step in here. So the RNA, instead of going straight to polypeptide, in eukaryotes, we do RNA processing. Then it's translated into the polypeptide. And specifically, if we looked at that, we basically talk about turning pre-RNA into full transcript mature RNA and taking that mature RNA and then reading that. Some important players in the translation process, we said RNA polymerase. There's a number of components, but tRNAs are involved here and they're important. They help bring in amino acids, the rRNAs, which are part of the ribosomes. And there's many other parts, but those are some of the most important that we're going to discuss in our basic understanding of translation. So here's the overall look at the transcription, translation, gene expression process. Okay, let's look at two different cell types, the prokaryotic and eukaryotic, and see how these processes of transcription, translation, RNA processing fit into them. Okay, so here is going to be, I'll draw the prokaryotic cell first. Prokaryotic cell, no nucleus, so all this is happening in the cytoplasm. We'll write that in a minute. So we have our DNA, and actually the DNA is circular and still double-stranded. That DNA is red, and parts of it are red, and they make RNA. And then that RNA is read by little ribosomes to make polypeptides. So if we look at the cell parts, we have number 1, and just go down. Okay, so number 1 here, that's going to be the cytosol. That's where all these processes happen in the prokaryotes. Then we have our number 2 process, or number 2 molecule in this case, that's our DNA. Number 3 is the process that takes DNA to RNA, that's transcription. Number 4, that's the mRNA. Number 5 is translation. Number 6, those are the polypeptides. Number 7, those are the ribosomes. If we looked at polarity here, we do need to know this as well. The 5' end, 3' end of the RNA. And that's just important because that tells you where the ribosomes start and where they end. You could also add in here the polypeptide ending up being folded into a functional protein. So we could call that 8. Enable 8, our protein. Maybe it comes together with another subunit that's also been transcribed from a different gene. Finally, we could also be specific about the kind of molecule that is doing number 3 process, the transcription. So we can name that number 9. Number 9 would be the RNA polymerase. It's going to read the DNA and make the RNA from the DNA sequence. So that's prokaryotic process of transcription, translation, gene expression. Now let's look at eukaryotes. So the difference here, well, one of the differences is that there is a nucleus. And our DNA is located inside the nucleus. This time it's in linear chromosomes. Just draw one of those there. Okay, so let's go through the parts here. So number one, where's that? That's the cytosol. So you can see that some processes happen there. Number two is the nucleus. Other processes occur there. Then we have number three, that's our DNA molecule. Number four, the process of transcription. Number five, that's our what we're going to call pre-mRNA. Number six is RNA processing, which includes putting caps on the end. That's what this little dot is here. It's putting a 5' cap, poly A tail, many adenines on the tail. And you see it's also shorter than it used to be. So the pre-mRNA gets parts of it, chunks of it spliced out, which means cut out, and the rest of it's put back together, because that's called splicing. Number seven, that's our mature RNA. Number eight process there is the translation. Number 9 is the polypeptide. Number 10, those are pointing to the ribosomes. Number 11 is our processed and folded protein. Just like in the prokaryotes, we could be specific here about what molecule is involved in taking and doing the transcription. That would be number 12. 12 would be RNA polymerase. So you can see in this case of eukaryotes, the transcription and processing happens in the nucleus, where the translation and then the protein processing happens in the cytosol. So how does this transcription translation process generally look? Here's our double-stranded DNA. You can see A, C's, T's, and G's are the nucleotides, adenine, cytosine, guanine, and thymine. Antiparallel strands, 3', 5', 3', 5'. And one of the two strands is going to be the template. For every gene, there's a specific template strand, but different genes use different strands. So in this case, the top strand is our template strand. We're going to use that strand and build our RNA off of it using base pairing rules. So just like when we build DNA, A went with T and C went with G, it's going to be the same kind of idea. There's specific RNA nucleotides that go with DNA nucleotides, and that's our transcription. You'll notice that here's our big DNA molecule, and parts of it are what we call genes. Those are just DNA segments, different sequences that actually code for a protein or functional RNA product. There are parts in between the genes that are not coding. Now, they don't do nothing. There's different functions for them. Most of which we don't know, but the gene parts are the parts that code for the protein. So let's figure out what our RNA would be. Okay, it's complementary base pairing with the top strand here. So A in the DNA is going to match, in this case actually, with what we call U or uracil. So there's no T in the mRNA or any RNAs. RNAs have U's instead of T's, but everything else is the same, minus the fact that it's an RNA nucleotide with ribose sugar rather than a DNA nucleotide with deoxyribose sugar. So here we have a C for the DNA, which is a G for the RNA. Again, here's another A, goes with U, U, U. Some more C's going with G's. Here's a G that goes with a C, A, U, G, C, and then T goes with A. So that's our RNA strand from our DNA template strand. And you'll actually notice that it's the same as the non-template strand. except for instead of thymines, there are uracils in the RNA, but the sequence is the same. So that's how we make our RNA molecule. That RNA molecule then gets read, and it's going to code for specific amino acids, which get strung together to make a polypeptide. So here's the key of how DNA has so many different combinations. So it's not just read one base to one base. If you just read uracil and uracil coded for one amino acid, all you'd end up having was 4 different codes. So, your uracil would be amino acid number 1, your adenine would be amino acid number 2, your guanine would be amino acid number 3, and your cytosine would be amino acid number 4. We know that there are 20 amino acids, so this 1-to-1 code does not work. If you do 2 bases and you do all the combinations there, that's 4 to the second. 4 different choices looking at 2 at a time. So that gives us 16, which is not quite 20, and we need 20 different amino acids coding for. So it turns out if you go in groups of 3, that's 4 to the third, that gives you 64 different options, which is plenty for the 20 amino acids. This also means that there's multiple groups of 3 that can code for the same amino acid, and we'll see later why that's important. So we call those groups of 3s in the RNA as well as the DNA are called codons. And you have to read the RNA in the correct codon chunks, and we'll discuss how that's found out later. But if you look here, basically UGG is our first codon, UUU is our second codon, GGC third, and UCA fourth. And each of those codons, there's a specific kind of amino acid that gets put in. So in UUG, codon on the mRNA ends up with a tryptophan amino acid put in. Then that tryptophan is attached to the UUU coding for a phenylalanine amino acid. The GGC coding for a glycine amino acid, that's now attached covalently. And the UCA coding for the serine amino acid, also attached. And so you've built a little polypeptide chain. And that process is called translation. So we have charts and wheels that have the code on it, so you don't have to memorize what triplets are which amino acids. And so there's 2 different kinds of ways you can read these things. There's these charts, and the charts, you start on the left, and this is the first RNA base. And you can remember it's RNA, not DNA, because there's the uracil in the code. So let's say we have the triplet AAA, and we want to know what amino acid that codes for. So we'll go here, and this is in the mRNA. And we start with our A, then we go to the top, and we find our second A, and so that ends up with intersecting at this box, and then we find our third one, which is here. So that means an AAA codon in the RNA codes for the lysine amino acid in the polypeptide. You can also use this wheel to do it. You start in the middle and work your way out. So let's do the same thing. Here's our A, and then here goes to another A, and then our third A, and then that's lysine. So I'll give you the option of using either of these types of charts. You'll also notice that there's 4 different types of codons that are sort of special. AUG is a start codon, so that's where the sequence of codons starts. And then there's 3 different stop codons, which don't code for amino acids. They code for nothing, basically, which means that the polypeptide will be stopped being made there. When we look at the genetic code using those codon wheels and codon charts, it turns out that almost all organisms use the same basic code, from bacteria to archaea to humans, which are a type of animal, to plants, to fungi, to amoebas, and all kinds of algae. So what that tells us is common ancestry. It's a really nice example of common ancestry evidence that the original organism that gave rise over a long period of time to the diversity we see today used that code, and it was successful, and so the organisms today also all use it. Now, different ones have certain bacteria might have a slightly different amino acid they put in, and so that's been, along their line of evolution, something new that's popped up. But generally, you can use the code for everything. And what does this mean? This actually is quite useful for us in science, because it means that we can use genes from other organisms and put them into a different organism, and they'll be able to express them. What do I mean by that? So I mean, for example, this is a mouse here, which is a mammal, a type of animal, and you can see it's glowing green in certain light. And the reason it does that is because in its genome, there is a jellyfish gene. It's called the green fluorescent protein, GFP. It was found in jellyfish, and under conditions it fluoresces green. And so it's not just because we want to make a green mouse and, oh, isn't that fun? We can actually do really important stuff with this, as in this mouse embryo here. So the GFP is turned on only in certain places where other proteins are turned on. So if you take protein A and put the GFP gene at the end of that coding sequence, whenever protein A is made, GFP is made. So wherever protein A is, GFP is being expressed too. It'll be green. So you can see that wherever protein A was, apparently it's in all this extra embryonic tissue here, because that's where the GFP shows up. Now we can also use different variants of GFP, for example, RFP, red fluorescent protein. So let's say we attach that to the back of protein B. That means that everywhere protein B is expressed, RFP comes up. And you can see that all the protein B is expressed all over the embryo. So this is the embryo of the mouse. So that can be very useful because sometimes it's, very specific cell places that different genes are turned on, and we can visualize that even in with movies in real time for externally developing organisms like a fruit fly. And we can see those genes turning off and on. We can also use this in order to use bacteria to make large quantities of proteins for treating human diseases. For example, a disease where you're missing a certain enzyme. It used to be that you had to harvest the enzyme from some other organism, like a pig or something like that, to use in humans. Now what we can do, though, we can just take bacteria, or yeast in some cases, put the human gene in for the enzyme that you are missing, and the bacteria will express that. And then you can just harvest the bacteria, purify the protein, and now you have the enzyme you're missing. It can be injected into the people that are affected by the disease, and it can help them live healthier, longer, less painful lives. So let's look at a real example of the different DNA all the way to protein. So here is from a database, the sequencing databases, hemoglobin, specifically the beta gene, the HBB. This is the DNA nucleotide sequence, and so here is our chromosome number 11, and if we look here, these three are different DNA sequences, and we have different variants that are involved, that kind of thing. So then that's our DNA. So if we look at the sequence of DNA, it's actually like this. We can tell it's DNA because there are T's in there. So lots and lots of DNA nucleotides all in a row. Then you see I've highlighted some in red, and I guess we should mention that this is 1,606 nucleotides. So these red ones, those are the parts that are actually going to be used to make the protein in the end. The black ones are parts that are cut out. So if we look at the DNA, when that gets transcribed, not all of it gets transcribed, but we get transcribed to mRNA and then cut up, and so put back together. That's the RNA processing. And so what we end up with here is the RNA sequence. And now it has T's in it. It's because that's the notation. But if we were really looking at RNA, it would have U's. So here is our RNA, mRNA, and here are the parts that get cut out. And so we have these three parts that are put back together, which give us this sequence. So now we're down to the pre-mRNA, RNA, 444 once it's processed. So then the RNA that was transcription is taken and translated into a protein sequence. So for every 3 nucleotides here, A, T, G, let's say, we put in a different amino acid. So in this case, put in methionine. And if we looked at the codon wheel, that's what it would show us, is that the AUG in the RNA is the methionine. And so you can see we have a string of amino acids now. Each letter stands for a different one. 147 amino acids. So we just went from 444 nucleotides down to 147. And then that polypeptide gets folded into a specific structure. This case, we're looking at the blue and orange subunits of the hemoglobin come from this gene. And you can see that they have all these alpha helices, and they come together in multiple subunits to make quaternary structure, along with these other subunits from a different gene. And then we have hemoglobin, which can carry oxygen through the blood and oxygenate the tissue. And so that's the basic process that you'd see going from, you can see a large sequence of DNA to a functional protein in the end.